All right, I think we're going to get started with the next session, which is composed of two plenary sessions. It's for me a pleasure to introduce Professor Michael Heneka from the University of Bonn in Germany. Professor Heneka studied medicine at the University of Tübingen in Germany and did a postdoctoral training at the University of Illinois in Chicago, and also at Cleveland at the Case Western Research University. Back to Germany, he was appointed as a senior physician in neurology at the Bonn University, and then he moved as full professor and head of the Department of Molecular Neuroscience at the University of Münster. Currently, he is full professor and director of the Joint Memory Clinic of the Department of Psychiatry and Neurology of the University of Bonn. Professor Heneka is one of the top leaders working on neuroinflammation in relationship with Alzheimer's disease and neurodegeneration, and has been, I would say, a pioneer in that area. He's published very relevant articles in the area, as probably many of you know, and his work has been widely recognized and received several awards. Professor Heneka has several excellent links with Spain. And I was just learned that he may be giving his talk in Spanish. Just kidding. But anyway, so we will be happy to have you, Dr. Heneka. He is speaking about does induced immune activation drive Alzheimer's disease. So Michael, welcome to Santiago, and thank you very much.
Miguel, muchísimas gracias, pero al fin me faltan muchas palabras, idiomas en español, palabras técnicas, y todavía no puedo dar una charla en castellano, pero el día viene. Thank you very much for this very, very kind introduction. And what I would like to do during this lecture is to point out some things which we may have overseen in discovering the pathogenesis of Alzheimer's disease. And at the end, I would like to discuss with you the question whether or not innate immune mechanisms may be a valuable target to change the course of this disease. So, like Alzheimer has pointed out, and for various historical reasons, we have focused on these initially described pathological changes, the tau phosphorylation and the extracellular deposition of beta amyloid. We have characterized them very well. Already Alzheimer himself did so, and we now have elegant stains for neurofibrillary tangle formation and amyloid beta. So how is this actually related to innate immune activity, and what is the innate immune system? So during our entire life, from our first days on, we're exposed to viruses, fungi, and bacteria. And the innate immune system is our first-line defense system to protect us. And if we like that as neuroscientists or not, the innate immune system had to make big steps in order to provide the body with the energy to develop a larger CNS. And phylogenetically, the big steps of innate immunity were ahead of the brain development. So, in other words, the reason we made it to the 21st century as a species very much resides in our capability to defend our body from bacteria, from viruses, and from fungi. And we don't have to go back very much. Just in the medieval times, human aging was very much restricted by the death reason number one, which were infectious. Now, the innate immune system has developed over millions of years, and it has been equipped with various surface receptors for all the bacterial components and viral components which have to be sensed by the myeloid cells, which actually represent our innate immune system. And here you see such a myeloid cell, a macrophage in the bloodstream, and we come back to that in a second. There are various surface receptors on these cells which can sense these bacterial components, and most of them signal to the MyD88 or IREC pathway in order to initiate inflammatory signal transduction. Now, I'll show you what this macrophage will do in a second. This is the macrophage, these are blood cells, and this is a bacteria. And now, this is what these guys are doing for us. They hunt the bacteria, they follow them, they sense them as long as they actually take them up and destroy them. So this is what your innate immune system does actually every day, every moment, even now while you're listening to this presentation. So there are various innate immune systems in the bloodstream, but there are also organ-resident immune cells, such as microglia, alveolar macrophages, copper cells, mesangium cells, and gut-blood barrier cells, bone marrow macrophages, and they all have an important role for protection from infection for these organs, but they also contribute to organ structure and to organ function. So this is a section of an Alzheimer's disease brain which is stained with Congo red, which shows you the beta sheet structured amyloid beta here, and you see the MHC class positive microglia cells surrounding or assembled at these sites. So what is the commonality now between a Congo red beta sheet structured amyloid and a bacterial surface? Many, many years before Congo red was actually used as a stain for human brain, it has been developed and licensed as a stain for bacteria, 1870 by Paul Bötticher in Leverkusen in Germany. And the reason was that it would actually stain beta sheet structured amyloid on the surface of many bacteria we know. So this is very elegant work by Matthew Chapman and colleagues pointing out in a science paper in 2002 that bacteria unexpectedly present beta sheet structured amyloids on their surface. And this is what was stained by Congo red. Now, in other words, this means that what these microglia are actually sensing with their surface receptors is a beta sheet structured amyloid, but to the microglia cell, it's as good as a pathogen invasion. It's a danger-associated molecular pattern. And these microglia cells have been educated via evolution for millions of years and equipped with these pattern recognition receptors to recognize these surfaces. And now they recognize the deposited amyloid beta. So they mount a very strong inflammatory reaction because, in their sense, they want to defend the body. What happens if microglia cells get activated? There is an inflammatory reaction which releases complements, cytokines, chemokines, nitric oxide, and radical oxygen species, so pro-inflammatory type of acute reaction. On the same time, these microglia cells stop doing very, very beneficial things they usually physiologically do for our brains, which can be summarized under surveillance function. So they have a reduced synaptic scaling function, they reduce their production of BDNF and various other trophic factors, and they stop clearing debris from the brain. This is a physiological microglia cell with all their branches in a high branch order and area covered, and these is the very same microglia cell activated at an amyloid beta deposit. And as you easily can see, has lost all its branches, and you can imagine that it cannot do the same job as this cell over here. Now, when is amyloid beta depositing actually starting? And we had heard about this in a very elegant talk right before. And I still want to repeat this. This is a sequential image of the very same person, patient suffering from familiar Alzheimer's disease, and we start minus 25 years prior to the cognitive deficits. So we see now that there is a constant increase in the brain of amyloid beta. Now we're minus 15 years. You see the precuneus, the frontal cortex, 10 years, Patient still doesn't know that he develops the disease. And now, when it turns 0, for the first time, he has cognitive deficits. And I want you to remember this multicolored picture. We'll get back to that at the end. So, first of all, this means that in one brain hemisphere in the very same brain region, we have various stages of amyloid beta deposition, and knowing that amyloid beta is such a strong danger-associated molecular pattern, this means that we also have different activities of the inflammatory reaction. But if we put this on a scale, and there's good evidence that this is also the same for the sporadic type of the disease, then the deposition may start as early as in the 4th decade of life. And knowing that it's just such a potent immune stimulant, we can conclude that from then on, this inflammatory reaction will influence all the other mechanisms which are ongoing in the brain of these patients until the very end of decline. And just to remind everybody, tau positivity, at least by PET means, starts to become positive when the cognitive decline sets in. So this is what we did know about innate immunity 20 years ago. And this was 2 years after I started working on inflammation in Alzheimer's disease. This is what we know today. And for the rest of my talk, I will focus on this part here, which deals with the so-called inflammasome complex, and in the case of Alzheimer's disease, with the NALP-3 inflammasome complex. So how is this NALP-3 inflammasome working? It requires binding to a toll-like receptor, which activates the MyD88 pathway, the mitosome, leading to NF-kappa beta activation and transcription of proforms of IL-1 beta and IL-18. At the same time, either amyloid beta itself or another immunostimulant can be taken up and resides in the lysosome. And lysosomal disruption, for example, or lysosome signals then represent an activation step which bring the NALP inflammasome into a multimer complex, which consists out of caspase-1, ASC, and NALP-3. And this complex then is active and converts the proforms. It activates caspase-1, and caspase-1 then cleaves the cytokines in their active form. And without caspase-1 activity and NALP inflammasome activity, there is no cytokine release because the IL-1 beta is the initiating cytokine for many others to follow. So when we looked in this inflammatory situation in the patient brain, and especially analyzed this system, we started looking to microglia cells, in this case in the frontal cortex of an AD case, and you see here this ASC-positive so-called SPEC. It's an ASC SPEC, and this antibody only gets positive when the NALP inflammasome is active. So this is a human microglia cell in the frontal cortex, showing that there is NALP inflammasome activation in Alzheimer's disease. And this active caspase can actually be graded, or the activity can be graded by blotting for cleaved caspase-1. Though this is a byproduct, a cleavage product, which is an activity signal. When we now compare AD cases and age-matched, non-demented controls, we see that the controls have almost no caspase cleavage activity, while the AD cases have a dramatic increase. And this is not only the case for Alzheimer's disease brain specimen, this also can be found in MCI due to AD cases. So there is a strong increase in caspase-1 cleavage, and by this, NALP inflammasome activity already in the stadium of mild cognitive impairment. So one important thing is, if we speak about innate immunity, I told you that the innate immune system has been developed phylogenetically over 1 million of years, and it's easy to conclude that it's different between model mice and humans, for example. So, but about 40% of our innate immune system is completely shared between mice and man, and in this case, the NALP inflammasome pathway, which consists out of PICARD, which is ASC, IL-1 beta, caspase-1, and NALP-3 is highly conserved. It's always expressed in microglia macrophages in mice and in man. So it's really, really completely the same in both species. Now this, in my view, allows us to study the innate immune system, and in particular this signaling transduction pathway in mice, and we did so, and we found that if we would knock down NALB3 or caspase-1 activity in several mouse models of Alzheimer's disease, we would reprogram microglia. So we would stop them from becoming activated. We would block inflammatory gene transcription. We would see an increase in A-beta in vivo phagocytosis, thereby reducing the cerebral amyloid beta load. We protect from spine loss, and most importantly, we protected from hippocampal LTP suppression, and we found that we have an almost completely protected spatial navigation and object recognition memory. And these are mice which are 18 months of age. So APP-PS1 double transgenic mice, which in the red line have no treatment, learn almost nothing in the Morris water maze experiment. They are almost completely protected in the case of NALB knockout or caspase-1 knockout. But what does this mean? This does by no means mean that this would be a good pharmacological target, because a genetic knockout starts from day one. So it's very, very early, and we never will have a patient situation where we can treat kids from their earliest days with any pharmacological drug which would prevent a disease that occurs maybe 70 or 80 years later in their life. So that was not a good indication yet. But as we have discussed before, a human brain does not show like this in a day or in a week, in a month, or even not in a year. The degeneration and the pathological changes take a long time, and importantly, there is spread of pathology within a brain region and from one brain region to another brain region. So there is a transport or a mechanism which leads to augmentation of pathology in these brains. And the classical view until very recently, I would say, was that you have the amyloid beta fibril, which is elongated through from a nucleation phase going into a growth and elongation phase until the fibril reaches a certain length and then fragments, and one of these fragments would be the new nucleus for the next elongation round. And our question was actually, does innate immunity contribute to the disease progression and, importantly, to the spreading of this disease? So the reason we asked that was a very old paper of ours, which we published together with Fred van Leuven from Belgium in collaboration, and Fred gave us a London mutated mouse overexpressing the APP-V717I mutant, and you can see that these animals don't have any extracellular amyloid pathology at 3 months of age. At 16 months of age, they show robust deposition. So they start at month 14, some even kind of some weeks earlier, but it's a late single APP transgenic mouse model with very slow amyloid beta deposition. So in these mice, very early, when we stained for CD11b positive microglia and GFAP positive astrocytes, we found focal microglia and focal astroglia activation. So way before any real extracellular deposit. And we wondered, what is the nature of this activation? So if we look by immunocytochemistry, we found that these cells would be all IL-1 beta positive, indicating that they had an active NALB inflammasome, because otherwise IL-1 beta would not come around. So we started to look in more detail to the earliest time points and the earliest interaction of the innate immune system with the AD-specific pathology. And it turns out that microglia cells are not a stable population. Humans turn over about 30% of their microglia over a year, and this is even accelerated in the case of pathology. Now, how do microglia cells die? They die in a very controlled way in the brain by pyroptosis in a case of inflammatory activation. And this is by the release of this ASC complex. So the ASC builds up the NALB inflammasome, builds up multimer complexes with outward-directed caspase activity to generate a high amount of IL-1 beta. And this forms a gasdermin D pore in the outer membrane, which then kills the microglia cell, a process which is called pyroptosis, very well described in peripheral tissues. Now, the ASC release is a very elegant method of the innate immune system to mount a rapid response, because a neighboring innate immune cell now can take up this spec, the so-called ASC spec, and has a ready-to-use inflammasome. It doesn't have to go through the cycles of transcription and protein buildup. It has a ready-to-use tool and can use it itself to generate these cytokines. Now, when we looked in AD patient brain or APP-PS1 transgenic mouse material, we found that we can detect ASC specs within microglia cells, as I showed you before, and outside in the plain extracellular space, and we quantified this. It turns out it's only present in AD cases, not in controls, and it's age-depending in the APP mouse model. Now, we started to do this and induce this experimentally by priming cells with LPS and treating them with ATP, another inflammasome activator, and we build up assays where we can visualize the single specs and we can quantify the amount of ASC specs being generated. So with this system, we wanted to actually analyze whether amyloid beta would be a strong inducer of microglial pyroptosis, and I tortured a number of students and postdocs in order to show that amyloid beta would be a fantastic ASC inducer, but it was not the case. They all failed. So I said, I'll do it myself, and of course I failed even better. But the only difference was I took a red-labeled amyloid beta, TAMRA-labeled amyloid beta, and I took a look in the microscope. So it turns out that we found once the ASC spec has left the cell, at the very same moment, amyloid beta would rapidly bind to the ASC spec. And I show you a little movie. I want you to focus on this cell up here, which will release an ASC spec in a bit. And you already see there is red amyloid beta binding to it. So the moment these ASC specs are released, under pathological conditions, amyloid beta very quickly binds to it. When we looked by EM measures to this, you see amyloid beta peptides alone, the ASC spec alone, and I hope you can appreciate that the ASC spec here looks rather like a magnet to the amyloid beta peptides. So we concluded that if an ASC spec would be released by a dying microglia cell and would meet an A beta monomer, it would be a bad marriage, so to say, and it would be the seed for amyloid beta deposition. Because remember, usually amyloid beta would leave our brain, either in the extracellular space or it would be locally degraded. Many of us have a constant, or we all have a constant amyloid beta production, but hopefully we don't build up deposits yet. So we started to look into the consequences of this binding. And this is an assay which is a co-seeding assay to investigate whether one protein induces the aggregation of another. And it shows you the conversion of the monomer to the aggregate over time. And you see in green, it takes about 6 hours to convert all the amyloid beta monomers in this solution into aggregates. And now we add increasing concentrations of ASC specs, and you see that there is a very fast shift to the left. So it now takes less than 70 minutes to convert 50% of the ASC specs versus 250 minutes in the absence of ASC specs. And if we check this out by gel electrophoresis, you can see that after 24 hours, we have very little aggregation in the absence of ASC specs, but in the increasing concentration of ASC specs, we see this conversion, and out of a sudden, we can detect oligomers and fibrils, but almost exclusively in the presence of the ASC spec. Now, this is amyloid beta 1 to 4D, and this is the very same with 1 to 4D2. So that tells us that ASC specs are relevant and strong drivers of amyloid beta aggregation. So we wanted to know whether this is specific or this is some unspecific effect, and we did a lot of control proteins and peptides, and turned out to be very specific, so we dived more into the molecular details of the ASC spec. We have a PYD domain and a CART domain here, and a high polar region by these lysine group up here in the PYD domain. So we exchanged the lysine to an alanine and generated a recombinant ASC spec. And first of all, the recombinant ASC spec in this case put together with amyloid beta 1 to 40 wild type shows the same reaction you see over time, over 72 hours. This is the aggregation. You see the A beta 40 in the recombinant aspects has a stronger aggregation, and more importantly, it generates oligomers and high molecular fibrils. So now we started to mutate these three high polar region-specific lysins, and it turns out that it's completely gone. So the aggregation, the interaction between amyloid beta and ASC is very specific and depends on the PYD domain of the ASC molecule. Now, another experiment done by Satish Kumar and Jochen Walter in the lab was this. So what we did over here, we did a co-sedimentation assay. We look at amyloid beta 1 to 40 and 42 alone in the presence of ASC or ASC alone. And what you see is here that all starts in the supernatant, and after 6 hours, it's completely found in the pellet fraction. In the case of amyloid beta 1 to 40, it's an all or nothing effect, and for 1 to 42, it's almost the same. Which means that in this in vitro experiment, we kind of induce an artificial plaque, and the ASC spec is able to track down the amyloid beta into a plaque completely. Now, it's more interesting to look after mice, so we are interested to look at the plaque situation in the mouse model, and we isolated these plaques and did a co-immunoprecipitation experiment. So we did an IP for ASC and blotted for amyloid beta, and you can see that in an age-dependent fashion, we detect amyloid beta bound to ASC. If you look to the plaque in more detail by immunohistochemical means, and we split the fluffy fiber compartment from the core, first of all, by sedimentation assays, we see that ASC and amyloid beta is present in the core, in the fiber compartment, and in the core compartment. In the immunohistochemistry, you can appreciate that the core is very much made out of ASC bound to amyloid beta, while the 6010 positive amyloid beta surrounds it. And this suggested to us that the ASC beta binding could be a seeding moment of this phenomenon. And for that, we turned to a model which has been introduced by Larry Walker and Matthias Jucker, and nicely described in this Nature paper in 2013. What they do is they compare transgene-driven deposition to induced deposition. They take lysates from 80 patient brain or APP transgenic mice, inject it into the hippocampus, and observe a strong spread of pathology. Now, after a certain period of time. This model has some weaknesses because synthetic amyloid beta is not seeding, which means that something is different in the brain lysates, right? So amyloid beta is either different itself, or there's a cofactor important for inducing the seeding and the deposition of amyloid beta. So I show you 2 out of 4 experiments, and this is the key slide of the entire presentation. So slow a little bit down. What we did is we used murine brain lysates derived from wild-type mouse brain or APP-PS1 mouse brain. And we injected this into the same mouse. One hemisphere received wild-type lysate, the contralateral hemisphere received APP-PS1. We did this in ASC knockout animals, in APP-PS1 mice, and APP-PS1 ASC knockout mice. What we did is we sectioned the entire mouse brain, and we did a histological quantification. All the sections had a defined distance to each other, and we took sections for histology and then sections for biochemistry. If we look at the comparison between the wild-type receiving hemisphere to the APP receiving hemisphere, we see that there is more plaque deposition in the hemisphere which received APP PS1 mouse brain lysate. And this is the open to the black bar. It's exactly the result Matthias Jucker has published in his science paper in 2009, and we verified this completely. If we now look to age-matched, non-injected mice, this is the gray bar. What you see is the effect of no injection, and you compare it to the wild-type mouse brain, you can see mouse brain injection itself has no effect. And now we go to the oligomer part. You see there is no effect on APP, no effect on beta C-terminal fragments, indicating that there is no effect on APP processing itself. There's a strong difference in total amyloid beta between the wild-type mouse brain and the APP PS1 mouse brain. This is a real strong oligomer band and fibril band, which is increased in response to APP PS1 mouse brain injection. And now we look at the mice which are ASC knockout. So just one inflammatory molecule. And this is the result. In the ASC knockout animal background, there is absolutely no increase of deposition. There is no increase in total area covered by A-beta or number of plaques. And most importantly, the ASC knockout mice, as in the pure in vitro experiment I showed you before, have absolutely no fibril and oligomer formation. And they have no increase in amyloid beta. That tells to me, at least for this mouse model, in this case, in this time course, that ASC's back formation is mandatory for amyloid beta deposition. So we have another proof, which is not in the manuscript, which was published last December. This is a setup where we use in vivo live cell imaging in the living mouse brain in non-anesthetized mice. These mice walk on the treadmill. They do a spatial navigation task, and they are under a 2-photon laser scanning in vivo microscope, so we can image calcium transients, for example, and other things which are fluorescent in these mouse brains. And in this case, we used an APP PS1 ASC knock-in M-cherry mouse model. So all the ASC specs which are present in this mouse brain are M-cherry positive, in this case red. And then we injected methoxyXO4, which is a similar compound to the Pittsburgh compound B, and it labels amyloid beta in blue. And I hope you can see that surrounding these ASC specs, we have a blue halo showing in vivo binding of the effect I just showed you biochemically. So the ASC spec, once released in the mouse brain of a living non-anesthetized walking mouse, is binding rapidly to amyloid beta. Now, the next experiment is how can we actually use this for a treatment strategy. And we did a very simple experiment. We used APP PS1 lysate injection with isospecific IgGs and an anti-ASC spec antibody and injected this into the respective brain hemispheres. And before we had shown that these antibodies have a strong anti-ASC inducing effect in this assay, while the isospecific IgGs have absolutely no effect and don't affect ASC-driven amyloid beta aggregation. Now, injecting this in the same hemisphere completely abolished the activation and the driven deposition. So this is the difference as before. This is the effect of the anti-ASC antibody injected at a single time point. Now, I'm a neurologist, like hopefully many of yours, because I think it's important that clinicians do basic research as well and bridge the gap in between both parts. And I'm interested what happens in my patients. So we took the very same assay, we isolated the plaques from the patients, and we again run a co-immunoprecipitation assay for ASC and blotting for amyloid beta. And we looked at about 30 human brains, and this is the difference. We never found a binding in non-demented age-matched controls and only detected ASC-bound amyloid beta in the case of Alzheimer patients. Very interestingly, when we look to the sucrose gradient, centrifugation gradient, and we looked at three different conditions in control cases, MCI patients, and AD brain. So this is a dot blot assay, blotting for ASC and amyloid beta in the fiber and in the core compartment. Now, what you see is that in MCI patients, in the core, there is ASC, very strong signal, and there is amyloid beta, very strong signal. The only difference towards Alzheimer's disease is that you get additional amyloid beta in the fiber compartment. Now, if I would ask you to paint such a plaque, how would that look like? I bet it looks like this. Every human plaque we have been detecting so far has a red ASC core and a green, in this case 6E10. You can use whatever antibody you have at hand. You will always find the core of the plaque being ASC positive, and you will find the surroundings being A-beta positive. And that suggested to us that the seeding effect is really true, because the ASC only can get into the center if it's there first. Now, how specific is this for Alzheimer's disease? I cannot answer that question, but we had the chance to look at brain material from vascular demented patients, FTD cases, corticobasal degeneration, and AD cases, and we only found the A-beta-bound ASC in the cases of Alzheimer's disease. Now, is this new? No, it's absolutely not new. If you go back to paintings, I call this Microglia Armageddon. I don't see any living or healthy microglia cell in this picture. It's stained for ASC in red and green are the microglia ABA1 positive innate immune cells. Sometimes you see the ASC speck outside the microglia. Sometimes they just seem to be inside the membrane. So this is a scene of dying microglia cells. And this is the frontal cortex of an Alzheimer patient. If you look to the paintings by Alzheimer and Nissl, more than 100 years old, you find here exactly these pictures which depict microglial pyroptosis. And you can find many of these figures back in this scene. So it's not new. We just had to re-find it again. Now, but it doesn't solve an important question. We have amyloid beta deposition very early now. We accept that we may have amyloid beta deposition in the middle of our life and not at the end, which makes Alzheimer's disease not a disease of the aging population. It makes Alzheimer's disease or a disease or disorder of the population, which is our workforce in their 40s, 50s, and 60s. Now, tau comes up very late. And if you trust the tau PET studies, which are out now, tau starts with the first decreasing mini-mental state examinations and an overt cognitive decline, maybe a little earlier. And the question is, what is the role of innate immune activation here? Or is there any role of innate immune activation? It's not given. Now, tau phosphorylation, and I hope you forgive me, but this is a simple cartoon. There is normally the tau protein stabilizes the microtubules. There is destabilization by phosphorylation of the tau at various sites. Phosphorylated tau then doesn't bind to the microtubule anymore, and paired helical filaments arise when tau phosphorylation is not dephosphorylated by phosphatases, most prominently PP2A. Now, there are various kinases driving phosphorylation, and there is PP2A antagonizing this. And actually, tau phosphorylation itself is not a bad thing. All the animals which hibernate are phosphorylating their tau to turn down activity, because the brain is the organ which consumes most of the energy. And if you sleep over the winter in some cave, you may not need so much brain energy, but you may want to survive and come into the next spring. So once dephosphorylation starts in the spring, you also find the end of the cage back, and you can hunt again. So in the case of the polar bear or some other hibernating animals, this has been shown that this is a very elegant balance here, actually. So in any case, when the kinases supervene and the phosphatase activation has no balance to it, then neuronal death sets in. So as a mouse model, we use the so-called Tau-22 mouse generated by Luc Bouet from Lille, and we believe that this is a good model because if we compare tau or neurofibrillary tangle formation in man to what we see in these mouse models, this looks pretty similar to us. So we decided to cross this mouse into this tau mouse into the ASC knockout mouse or into the NALP3 knockout animals. And then we waited for 8 months, not having a lot of experience with these animals, to be honest, but we were in collaboration with Luc Bouet, who has generated the mouse. And what you can see is that at 8 months, this is the tau transgenic mouse showing pathology in the CA1 and CA2 region, maybe mostly CA1. Then you have the ASC knockout, which shows a clearly decreased pathology, and the NALP knockout, where pathology is almost absent. So this is a very strong decrease of tau pathology in the hippocampus or in the CA1 region. And I was very unhappy about this result because I don't want to get a name in science for rescuing everything with one single pathway. This is a stupid thing. This cannot be even true. So I exchanged the whole team. I got a new postdoc. I got a new technician. They would not know from each other, and I waited longer. So I didn't trust the 8-month analysis. I thought maybe some of these animals were delayed in pathology. And then these people came back with this result at 11 months. So it's an independent cohort of animals. It's even another animal house. It's another group of investigators, and they show me the same thing. NALP completely or almost completely blocks tau-induced pathology. So what does the innate immune system do with the tau pathology cascade? We first verified this by looking at 88 and 8100 by Western blot and in sarcosyl insoluble tau fractions by Western blot, not an easy task. But we finally showed that there is substantial reduction in the load, especially in the NALP knockout, not so much in the ASC. And then we found that we actually modify the PP2A, the demethylation of the PP2A subunit C, which is responsible for neuroprotection. And the dephosphorylation here is increased by the protective dephosphorylation, so to say, is increased by the NALP knockout. The pathological phosphorylation at the same time is decreased through action on alpha-chem kinase 2 and GSK3 beta. So it's a triple action. The phosphatase activity is going up, and the kinase activity is decreased by NALP knockout and a little bit by ASC knockout too. And we turn the balance towards the phosphatase activity here, which then enables the neuron to survive. Now, we have very little immune reaction, but we have some. And we are currently looking by IMARES and an artificial intelligence guided program, a learning program which scans the entire mouse brain and takes the arborization of these microglia cells. What you can see is that you have activated rounded microglia cells. In the case of the NALP knockout or ASC knockout, you have more microglia branches, and as a sign of reduced inflammatory activation. Now, ultimately, it's all about behavior and function, because we wanted our patients to get better or not to get cognitively impaired at all. And this is another example of the Morris-Water maze test. But in this case, we are not looking at these 8 days of learning. We are more interested in keeping information. So we look at day 9, when we take out the platform and the mouse have to refine the place the platform had been the previous 8 days. And they only can orient themselves at these internal cues. Now, this is a mouse which obviously goes very quickly to the platform. This is a mouse which is completely confused and seeks a long time until it finds the platform by chance or not at all. Now, looking at these animals, we find that wild-type, ASC knockout, NALP knockout animals all do fine. They spend much more time in quadrant 1, where the platform is, as in mean of the other quadrants. The tau transgenic mouse is a completely confused mouse. It doesn't remember anything about the platform position. And the ASC and NALP knockout mice do much, much better. They basically look like the wild type or the ASC and NALP knockout itself, despite their having the tau transgene. Now, in summary, I tried to make the point that beta-sheet structured amyloids are a known danger signal to myeloid cells, including microglia. If they find a beta-sheet structured deposit, they react as if they would see a true pathogen, because it's the same receptors and the same signaling pathway which is being activated. During various waves of pro- and anti-inflammatory changes, several danger-associated molecular patterns arise in the AD brain, but also in various other neurodegenerative diseases. In the NALP3, inflammasome activation is strongly increased. I showed you the activity signal in brains of Alzheimer's patients and MCI patients. ASC specs are being released by macrophage and microglia pyroptosis. It's a very defined dying pathway. ASC specs bind to A-beta peptides and increase their propensity to aggregate in a concentration and a time-dependent manner very strongly. And if the animal data are correct, they're the beginning and they're the true reason why amyloid beta deposits in the brain at all. The NALP inflammasome activation drives tau pathology and accounts for tau-mediated spatial memory deficits by modulating phosphatases and kinases in the opposite direction. For those of you who are interested in neuroinflammation, I would like to invite you to this school, which will be held in October in Conil de la Frontera in the south of Andalusia. We have a very eminent and very good experienced faculty. And if you want to come, I think there are still some places open. Please drop me an email. I thank all the people which helped to do this work, especially Carmen Venegas Maldonado, who is the first author on the 2017 Nature paper, but also Angelica, Grieb, Francesco, Jochen, and Satish, people from my lab at the University of Massachusetts, and of course, all the people who funded that work. I thank you for your attention, and I truly hope you have questions.
Michael, congratulations. Very nice presentation, very didactic and very appealing. I have a question. Do the regular anti-inflammatory drugs, do they inhibit microglia?
The regular?
The regular anti-inflammatory drugs, do they inhibit microglia? If it is true, why don't we use as a preventive treatment for Alzheimer's disease anti-inflammatory drugs? Okay.
So anti-inflammatory drugs have been proposed and have been used, but in my view, in a kind of senseless way. Because first of all, they've been used in very advanced patients, and then the assumption was that non-steroidal anti-inflammatory drugs like ibuprofen and indomethacin, who have approximately 280 known pharmacological targets, would work through inhibition of prostanoid synthesis. When we then had the prostanoid synthesis inhibitors of cyclooxygenase 2, like kelecoxib, rofecoxib, those drugs were tested in the AD patient cohort, and those drugs failed because prostanoids are only one single signal in the whole inflammatory cascade. And this is the only thing they inhibit. The rest of the inflammatory activation are not affected at all by these drugs. So I think we have to go at the very start of the innate immune pathway, or we have to be more selective in the innate immune pathways we want to modify in the brain.
Okay, thank you.
Any other question?
Nice. I was wondering whether it's known whether how the rate of pyroptosis by microglia develops over time.
That's a very good question. So the problem is that we would have to distinguish the rate of pyroptosis under physiological conditions, which is very low. There are two papers, one by Yuka and one by Prince. They suggest that over a period of 6 months, 13% of microglia would get lost. This comes up over a year to 26%, which is an interesting number, since the Karolinska Institute had a recent study showing that microglia in the human brain, in the living human brain, would turn over by about 30% per annum. So the animal, again, the animal data are not very far from the human brain data. I'm not sure, and I cannot claim that all these microglia would die from pyroptosis. There might be other cellular pathways where microglia decays, but in the case of, in the presence of an inflammatory stimulus, at least the delta of death of microglia cells seems to be due to pyroptosis. So the accelerated turnover in both directions, death as well as proliferation, seem to be, at least on the death side, due to pyroptosis. I don't have clear numbers for distinct ages, but it's a very good question.
All right. Thank you very much, Michael, for a very nice and stimulating talk.